going to start this plenary session on neurobiology of cannabinoids and therapeutical cannabis use. Before we start, I would like to remind you of something. You know, neurons using conventional neurotransmitters, serotonin, dopamine, GABA, acetylcholine, are neurons which synthesize the mediators and in the terminal part. And if they need, they can synthesize immediately. So there's an immediate synthesization. When you need dopamine, there's a local synthesis on the spot. Now, when you need to synthesize a neuropeptide, which is composed of amino acids, first and foremost, you need to capture amino acids, but you need also mRNA, transfer RNA, and this can also happen in the cerebral body. And if you do not synthesize the neuropeptides or their precursors, slowly they are going to move to the termination, and most of the time those neurons are pretty long. And so there's a period of time between the synthesis in the cerebral body and the moment those compounds are available at in the terminal part of a neuron. I'm explaining this, that if you ask a neuron using a neuropeptide to increase their respective activity, they're going to be depleted very quickly. However, there's a new pharmaceutical avenue that we have started to research. Once the neuropeptide is released to act on the postsynaptic bioreceptor, it has to be destroyed for the signal to be relevant. And so there's a new therapeutic approach through which we are going to block the degradation pathway of those neuropeptides for them to be maintained for a longer period of time on the synaptic part for it to get a synergetic effect. So it applies to cannabinoids, but also for the opioid peptides. So I wanted to make that clear because this is a new avenue for research, especially for opioid peptides and cannabinoids. And so it creates new avenues. Now, maybe it will take as long as 10 years to get the molecules to be used in clinical practice. Now, without further ado, I'm going to give the floor to the first speaker, Ms. Reem SMOUM JAOUNI, and I'm sorry if I mispronounced your name. She gave us an overview. I think that was on Tuesday. She gave us an overview on the past, present, and perspective of cannabinoids. But today, she's going to present results that are more centered around the role of cannabinoids on anxiety, nausea, depression, and addiction. Reem, you have the floor. Thank you.
Thank you. Good afternoon. Today I'm going to talk about the role of the cannabinoids in nausea, anxiety, depression, and addiction. Now, cannabis sativa, it contains a lot of bioactive compounds. And of these, we have cannabinoids, we have flavonoids, and we have terpenes. And the main representative of the cannabinoids, we have the tetrahydrocannabinol, which is, as I mentioned yesterday, it is responsible for the psychoactive effect of the plant. Now, other main cannabinoids, we have the tetrahydrocannabinolic acid, we have cannabinol, cannabidiol, and cannabidiolic acid. Now, the cannabinoids, when they are biosynthesized in the plant, they are biosynthesized as acids. And the main biosynthetic precursor of the cannabinoids, it's cannabigerolic acid, which is the main biosynthetic precursor for cannabidiolic acid, tetrahydrocannabinolic acid, and cannabichromanic acid. Now, these can generate their decarboxylated counterparts under the action of heat, so that cannabidiolic acid, CBDA, it will give CBD, THCA will give THC, cannabichromanic acid will give us cannabichromane, and cannabigerolic acid will give us cannabigerol. Now, most of the research has been done on the CBD, and there is very limited research which is done on the CBDA. Now, the CBDA is the major constituent of the plant cannabis sativa, and it has been isolated in 1955, and its structure was elucidated in 1965. And this compound has been shown to have an anti-nausea effect, to inhibit breast cancer cell migration, and to have anti-anxiety effect as well. Now, however, this compound is unstable, and its instability, it weakens the case of developing it as a medicine, and therefore we want to stabilize it. And if we want to stabilize it, we thought of comparing it with its CBD methyl ester, this compound. Now, the decarboxylation of the CBDA, it proceeds through the formation of carboxyl anion, and this is not the case with the cannabidiolic acid methyl ester. And indeed, we found that the cannabidiolic methyl ester, it is stable in the lab under different conditions, 4 degrees, -4 degrees, and room temperature under different circumstances up to 6 months. So we wanted to see the role of CBDA methyl ester in nausea, anxiety, and depression, and a little bit of what is known about the role of the endocannabinoid system in nausea. Now, the sensation of nausea is one of the most debilitating human experiences, and the current anti-emetic therapies, they are less effective in acute nausea, and they are completely ineffective in anticipatory nausea. And preclinical findings suggest that Cb1 receptor agonists, FA and MAGL inhibitors, which elevate anandamide and 2-AG respectively, the CBD and the CBDA, they have effect on reducing the acute and anticipatory nausea. And therefore, together with Professor Roger Pertwee in Scotland and Linda Parker in Canada, we were able to see that the CBDA methyl ester was much more potent than the CBDA itself and the CBD in enhancing the serotonin 1A receptor activation. And therefore, it was much more effective in reducing the acute nausea and anticipatory nausea-induced effect in the rat capping model. In addition, there is a complex effect of the internal and environmental factors on the endocannabinoid system, modulating mood and behavior. And what is known in the literature about the effect of the endocannabinoid system in anxiety and depression is very limited. However, we know that the effect of CBD, there is effect of CBD in reducing anxiety and depression, and there is an effect as well of marijuana on the PTSD. Therefore, we wanted to see the effect of the CBDA if it affects the serotonin 1A receptor. And we know that the effect of the serotonin 1A receptor on, there is a very strong effect of this receptor on treating depression. We wanted to see the effect of this compound on depression. And just shortly, I will discuss I will explain the mouse model that we are using. Here, the mouse, they experience a kind of stress. We named it food shock stress. And 24 hours later, they are given vehicles, CBDA or CBDA methyl ester. 45 minutes later, they are placed in a dark compartment. And the time that they spend in the light box between the two compartments is tracked. So if they spend time in the light compartment, then they do not have anxiety. If they spend time in the dark compartment, then they do have anxiety. And therefore, what we saw that the food shock stress, it greatly enhanced the time that is spent in the dark compartment, meaning that the rats, they have anxiety, and the CBDA methyl ester at a very low concentration, it reduced this time, meaning that it has a strong anti-anxiety effects. In addition, we wanted to see the effect on depression, and what we saw is that this compound as well, it has strong antidepressant effect at a very low concentration. The mouse model that we are using is the forced swimming test, and the mice, when they are depressed, or the rats, when they are depressed, they have high immobility and decreased swimming. And the CBDA methyl ester, it reduced the immobility and increased the swimming. So in order to summarize this part, the CBDA methyl ester, it displayed greater potency than the CBDA and the CBD itself in suppressing the signs of both the acute nausea and anticipatory nausea and stress by affecting the 5-serotonin receptor-dependent manner. And as well, the CBDA methyl ester, it has very high potent antidepressant effect. And the CBDA methyl ester, if it is an agonist for the serotonin 1A receptor, then any effect that it is, any pharmacological disease that is affected by this receptor, then this compound may be active against it. For example, cerebral infraction and pain. Now, the role of the endocannabinoid system in addiction. Now, the substance use disorder is a global problem with over 30 million individuals estimated to have SUD. And the endocannabinoid system, it plays an important role in the neurobiological processes involving SUD and by mediating rewarding and motivational effects of substances. And we have rimonabant and abuximols, they have Sativax, they have potential for pharmacological treatment for substance use disorder, and as well, CBD proved to be effective in reducing the SUD. Now, the main two endocannabinoids that are endogenous, they are anandamide and 2-AG. And these two endogenous endocannabinoids, they proved to have key biological activities in a lot of diseases. And besides 2-AG and anandamide, there are omega-6 fatty acid compounds that proved to have cannabinoid properties. And they were listed among the endocannabinoid family. They are noladiene ether, virodamide, and arachidonyl dopamine. Now, besides the endocannabinoids, the classical ones, we have the endocannabinoid-like compounds. Now, endocannabinoid-like compounds, they represent a major group of endogenous lipids. They are found in the brain and part of them in the peripheral nervous system. And they actually do not bind to the cannabinoid receptors, but their effects are cannabinoid receptor dependent, and they have numerous physiological functions. From these, we have palmitoylethanolamide that proved to be anti-inflammatory, anti-nociceptive, neuroprotective, anticonvulsant. We have oleoethanolamine that regulates food intake. We have arachidonyl serine, which is vasoactive and neuroprotective. And we have oleoyl serine, which proved to be anti-osteoporotic agent. Now, there was a paper that was published in Science in 2007 that, where the researchers stated that cigarette smokers with brain damage involving the insular cortex display cessation of tobacco smoking. And this implicates that this region in the brain is important for nicotine addiction. And in collaboration with another 3 groups in Canada, Richmond, Virginia, and Italy, and with the aim of finding compounds that may help treat nicotine addiction, we thought of finding neurochemicals in the insular cortex of the brain that were affected by traumatic brain injury. And indeed, lipidomic analysis proved that there is a difference between traumatic brain injured mice and the normal brain. There was an increase, a substantial increase in the amount of the endocannabinoid-like compound olylglycine in the insula. Of course, we looked at other compounds. We looked at endocannabinoids and endocannabinoid-like compounds, and of all of these, we found that only olylglycine had substantial increase. So in order to see the effect of olylglycine on the rewarding effect of nicotine withdrawal symptoms, this is the experimental design. We have mice that they were given nicotine or saline for 14 days. Then after 14 days, they are given olylglycine, several doses or vehicle. 15 minutes later, they are given the antagonist mecamylamine. 25 minutes later, they are tested for the elevated plus MACE test or the physical somatic withdrawal signs. Now, the elevated plus MACE test is a test that is used to assess the anxiety-like behavior in mice. And in this test, if the mice spend time in the open arm, then this proves that they are not anxious. And if they spend time in the closed arm, this proves that they are anxious. And what we saw that the mice that are nicotine dependent and they are undergoing precipitated mecamyl withdrawal symptoms, they have reduced time in the open arm, which means that they are anxious, and olylglycine, it increased the time in the open arm, meaning that it reduced the anxiety-like behavior in these mice. In addition, olylglycine reduced the mecamyl precipitated somatic withdrawal responses in the nicotine-dependent mice. And these, they are represented as paw and body tremors, head shakes, backings, curls, etc. Now, as well, olylglycine, it reduced the rewarding effect of nicotine. And this is tested by the conditioned place preference test. In this test, the mice, they are put in 2 neutral environments. Here, they are white and black shaded, and separated or spatially and temporally with unconditioned stimuli. For example, on days 2, 4, and 6, the mice were given drug injection in the black compartment, and on days 3, 5, and 7, they are given saline injection in the white compartment. And on day 8, they are given the opportunity to move freely between the 2 compartments, and the time spent in each compartment is an index of the reinforcing effect of each unconditioned stimuli. And what we saw is that olylglycine dose-dependently inhibited the nicotine-induced conditioned place preference. Now, olylglycine by itself, it didn't bind either to Cb1 or Cb2, but it inhibited Fa, which means that one of the mechanisms that is suggested is that it increases anandamide levels in the mice, and they behaved as well as PPAR alpha receptor agonists. Now, we wanted to see the effect of on morphine addiction as well. So what we saw is that olylglycine, it blocked the aversive effect of morphine withdrawal, and this was as well inhibited by the Cb1 antagonists. In addition, the mice that are undergoing morphine withdrawal symptoms, they have nausea-like somatic behaviors, seen as mouthing movement, abdominal contractions, diarrhea, and lying on belly. And oleoglycine attenuated as well these nausea-like somatic behaviors. The effect was prevented by the Cb1 antagonists. Now, oleoglycine has an amide bond. And this amide bond, it can be hydrolyzed in the body by amidases. So we thought of stabilizing this amide bond, and we thought about stabilizing it by introducing methyl groups on the adjacent to the amide bond. This has been done previously with anandamide, a compound called methanandamide. It has been in 1994. They introduced a methyl group, and this compound is much more stable than anandamide, but it has the same activity, but it is more stable. And therefore, we synthesized the methyl oleoglycine, introducing 1-methyl, and dimethyl oleoglycine, introducing 2 methyl groups. And what we saw is that the monomethyl oleoglycine, it produced a longer-lasting interference than oleoglycine with the acute naloxone-precipitated morphine withdrawal, which means that when oleoglycine is given 10 minutes before the naloxone, it gives an effect. But if it is given 60 minutes before the naloxone, it loses its effect. However, the methylated compound, the more stable compound, if it is given 10 minutes or 60 minutes before the naloxone, then it will have its effect. And this compound as well, its activity was prevented by PPAR alpha and Cb1 antagonists. And to summarize, the endocannabinoid-like compound oleoglycine, it interferes with the nicotine addiction and acute naloxone-precipitated morphine withdrawal. And the monomethylated oleoglycine, it may be more stable agent than the oleoglycine itself to combat the aversive effect of the acute naloxone-precipitated morphine withdrawal symptoms. And finally, I would like to thank our collaborators. We did this together with Roger Pertwee in Scotland, Linda Parker in Canada, Aaron Veller in Israel. We did depression with him. In USA, Aaron Lichtman, and in Italy, Vincenzo Di Marzo. And thank you for your attention.
On peut peut-être poser quelques questions et après on fera
So maybe we're going to take a few questions from the public, and then we'll have a further Q&A session afterwards.
I'm trying to understand the mechanism by which the oleoglycine could both decrease nicotine withdrawal and its acute rewarding effects. I could understand why raising anandamide levels might help with decreasing withdrawal, but it seems strange that it could also decrease the rewarding effects of nicotine, because rimonabant does that.
Yes, we have to look this further on. I mean, it's not, we didn't go further in these studies, but yeah, we will.
Yeah, just another question with regard to the rewarding effect, because it's really something is very surprising. No? Did you evaluate because condition pre-specified is just a condition response related to reward. Did you really evaluate reinforcing self-administration?
No, we didn't do self-administration. Still to be done. I mean, all these studies are preliminary. We have published part of them, but still we will do more. Yeah.
Just a brief question. Methylation of the anandamide is to protect anandamide from degradation by FAH? Ah, okay. It's only okay.
This has been done in 1994, and it was proved, yes, that it inhibits the degradation. Yes.
Bon, si nous n'avons pas de...
Right, if there are no further questions, but we might pick up on those very interesting topics later. The next presentation will be by Rafael Maldonado. He's a medical doctor, a researcher, a teacher at the University of Barcelona and University Pompeu Fabra. Last year, he presented an animal model which make it possible to study addiction to food. And in the last 2 years, he has compiled quite many results and is going to present the effect of cannabinoids on food addiction.
I would like to thank the organizers, and particularly Jean-Pierre, for giving me the opportunity once again to be here in this conference. So I will talk about the involvement of the endocannabinoid system in food addiction. And I will focus in one particular brain area, that is the cortico-limbic pathway, where this endocannabinoid system is very abundant. So, as you probably know, food addiction is a very old topic, but very controversial topic. Already in the 50s, 60 years ago, it was defined, it was postulated a possible food addiction as a concept that refers to some adaptive changes that occur in the brain only with some particular food, not with all kinds of food, and only in some particular individuals, not in all individuals. And under these conditions, these adaptive changes in the brain lead to behavioral alterations that mimic drug addiction. There is really a lot of common behavioral points in this food addictive behavior and in the behavioral alteration occurring during drug addiction. And in spite of, well, this is in the 50s, and today we have a very well-recognized food addiction score, the Yale food addiction score, that is used in most of the clinics just to define this food addiction, but it's a controversial issue because in the DSM-5, you don't have food addiction as a disorder. So it's still a controversial concept. So let's see first what is, very briefly, what are the neurobiological bases of drug addiction, very brief. Then let's see the model to evaluate food addiction, and let's see how we can use this model just to dissect the brain areas, the mechanisms involved in this addictive behavior promoted by food. Well, you know that drug addiction is now focused on, is now defined from a behavioral point of view. It's not like the DSM-3 that was a drug-centered definition. Today is a behavioral center definition, drug addiction. So drug addiction, DSM-4 and DSM-5 define this substance use disorder as a chronic relapsing disorder that is characterized by behavioral alterations that mainly are defined by 3 critical points. First, the difficulties to stop the use of the drug. Some drug addict is somebody that cannot stop the use of the drug. The extremely high motivation for the drug, really the center of all the activities of the drug addict is the drug, and the drug use is maintained in spite of the negative consequences. The addict knows the negative consequences of the drug, and in spite of these negative consequences, the drug is still used. These are the key behavioral points in this substance use disorder. Well, and why this substance use disorder occur? What are the neurobiological bases? Just in 2 brief details.
remember that drug addiction starts because all drugs of abuse are able to activate the reward circuit. This is the starting point. And this reward circuit, remember that we have an important area that is the ventral tegmental area that contains the bodies, the neurons that send dopaminergic projections to the limbic system. And all drugs of abuse are able to activate this reward circuit by increasing dopaminergic activity in the limbic system. The same occurs with food. Food is also able to activate this reward circuit. Well, this is the acute administration, the acute consumption of a drug. But after repeated consumption, there are adaptive changes in this reward circuit. And these adaptive changes led to a tolerance to the rewarding effects. The addict still feels reward, but not in the same, not the same height than at the beginning. But there are also the activation of other circuits in the brain, very important for the addiction cycle. Remember that we have the activation of all the stress circuits, so the addict must continue the use of the drug, because if not, you're going to feel very bad, because the stress circuit is going to be activated. There is also a change in the behavior. Just the behavior will be dominated by the dorsal structure, by the striatum. So the drug consumption will be just a habit. Not at the beginning, it was looking for the reward. No, at the end, the striatum plays a major role. It's just a habit that is impossible to stop. But probably the most important change, from my point of view, in the drug addiction cycle, occurs here, in the prefrontal cortex. In the prefrontal cortex, remember that the prefrontal cortex, medial prefrontal cortex, is the highest structure for the rational control of the behavior. And here there is a major change. Remember that the prefrontal cortex plays an inhibitory role on the limbic structure. So you control your behavior just inhibiting just these habits and responses that are mediated by the limbic system. And this control, this equilibrium between cortex and limbic system is crucial for a rational control of the behavior. And thus disappear during drug addiction. And for that, remember these very beautiful studies of Nora Volkow showing this was a cocaine addict, but for whatever addiction is the same. During addiction, you have a dramatic decrease in the activity of the cortical areas. So your capability for making a rational control of your behavior is disrupted. And you are unable to make an inhibitory control in your behavior. And this is probably the most dramatic changes occurring during addiction. So finally, during addiction, you have you start activating the reward circuit, but you have adaptive changes that make this behavior as an automatic behavior was an activation of the stress circuit and an impairment in your capability for control the behavior. And this is a very brief summary of a very long history. That is why we become drug addicts. And in this circuit, there is a particular neurotransmitters that control all these circuits. And this is a lipid, an endogenous lipid, that are the endocannabinoids. And endocannabinoids, these are the endogenous neurotransmitters. The two main endogenous are anandamide and 2-arachidonoglycerol. It was previously explained that are endogenous lipids. And they activate mainly two receptors, the Cb1 and the Cb2 receptors. Cb1 receptor is the most abundant 7-transmembrane domain receptor in the brain. There are a large amount of Cb1 receptors, so they must play an important role in the control of a lot of functions. And Cb2 receptors are also present in the brain, but they are mainly present in the cells of the immune system. So I'm just going to talk about the Cb1 receptor. Also, as a general idea, when you activate a cannabinoid receptor, you are mainly to produce inhibitory responses in the cells. You activate the receptor, you inhibit the cells. Just as a mnemotechnic rule. It's not exactly like that, but just to keep that in mind. So these cannabinoid receptors are very abundant in the brain. And we have in all these brain areas that are involved in the control of reward and the control of the behavior, you have a large amount of Cb1 receptors. Cb1 receptors are first located in the ventral tegmental area, where you have the cells body of the dopaminergic neurons. You also have Cb1 receptor in the limbic system, in the nucleus accumbens. And you also have a very good concentration, very high concentration of Cb1 receptor in the cerebral cortex, in the prefrontal cortex. So in every key structure involved in drug addiction, you have Cb1 receptors. And Cb1 receptors, in general, they are going to play a permissive role in order to obtain the release of dopamine. So remember, when you consume a drug, you feel reward because you release dopamine in the limbic system. To release dopamine, you need an intermediate that are the Cb1 receptors. And Cb1 receptors, mainly they are going to modulate the activity of two opposite neurons, GABAergic and glutamatergic neurons, activatory and inhibitory neurons. Through very complex circuits, they really play a permissive role in order that the drug will be able to activate the reward circuit to release dopamine. This is for drug of abuse. For food, it's the same. With food, when you consume food, you feel reward, of course. But in order to feel the rewarding effect of food, you also need the same intermediary. You also need the activation of Cb1 receptors. So they play a major role in drug addiction. We already saw in the previous presentation, but not only in drug addiction. We are going to see now what's happening in food addiction with these Cb1 receptors. So may we extrapolate all these systems all these circuits to the food addiction? This has been the aim of our studies this last year. So to study food addiction, well, to study food addiction, first thing, a model of food addiction, a behavioral model of food addiction. We do have a behavioral model of food addiction that I already presented last time in this conference. So in this model, to develop food addiction is not easy in animals. So it's a model a little bit complex. It's an operant behavior model. Operant behavior, remember, this is a Skinner box where the animal press a lever, and when the animal press the correct lever, the active lever, is going to receive a reward that is food. We have used, for this model, we have used two kinds of food. Chocolate pellets, that is the one where we obtain the positive result, and standard food, where standard food, I anticipate, we are unable to develop addiction. We need chocolate pellets to develop addiction. So what was the protocol? The protocol, we train the animals during 120 days. Very, very long training, very long training. In a fixed ratio 5. Fixed ratio 5 means that animal must press 5 times in the active lever in order to obtain a small food pellet. And we evaluate the addiction criteria at the beginning and at the end of the training period. And we use the addiction criteria defined in the DSM-5. So remember, these 3 addiction criteria is the difficulty to stop the use of the drug, the high motivation for the drug, and the consumption of the drug in spite of the negative consequences. How we can evaluate the difficulty to stop the consumption of food? What we do is as follows. We have a session, a training session, that the duration is 1 hour. But in this session, we have 3 periods. 25 minutes, active period. 10 minutes, the light is off, so the animal can do whatever, is not going to receive the food. And then again, 25 minutes of active period. So an animal that is unable to stop the consumption of food, you can imagine that during these 10 minutes that the light is off, the animal is going to persist and he's going to press once again, in order to obtain food, in spite that he knows that you are not going to obtain food. He's unable to stop the behavior. Motivation, how we evaluate the motivation for food. We use a protocol that is the progressive ratio protocol. To obtain the first pellet, the animal must press 1 time. To obtain the second pellet, the animal must press 5 times. To obtain the third pellet, 12 times, 21 times, blah, blah, blah. There is a moment that to obtain 1 single pellet, the animal must press 5,000 times. You can imagine the animal is not going to do that. But you can evaluate what is the maximum effort that the animal does in order to obtain a pellet. And the consumption of food in spite of negative consequence. How we can evaluate that? We use a punishment test. Remember that to obtain the food, animal must press 5 times. Animal must press 1, 2, 3, 4. When the animal presses the time number 4, receives a food shock, not the pellet. And when the animal presses the 5th time, receives another food shock, and then the pellet. So the animal must be really very crazy to look for pellets under these conditions, because it's 2 food shocks in order just to get 1 single pellet. So let's see what's happening in this model. When we use this model, remember that we have standard pellets, chocolate pellets, training at the beginning, training at the end. We obtain this addictive-like behavior, you can imagine, only with chocolate, not with standard food, and only at the end of training. You can see here the persistence to respond, how animals train with standard food, they don't persist, you turn off the light and that's fine. With chocolate pellets, at the late period, there are animals that press 600 times during 10 minutes, so 1 per second. They are unable to stop. Also in this period, you have animals, and animals trained with chocolate, that press more than 1,500 times the pellet, the lever, in order to obtain 1 single pellet. This animal, the motivation is extremely high. And animal with the resistance to punishment, animal that get 40 pellets with food shocks. That means 80 food shocks and 40 pellets. This animal forget about the punishment. It's looking for the reward, for food. So you can see here at the end that you have a dispersion of the results. There are some few animals that develop this addictive-like behavior. And here we use the same criteria that DSM-5. We use a criteria of addiction. So those animals that have 0 criteria, we define as non-addict. Those animals that have 2, 3 criteria, we define like addict. And you can see that in animals trained with standard food, no one was addict. However, in animals trained with chocolate pellets, there was 22% of animals that developed food addiction. So we are able to mimic this food addiction behavior in animals. So these animals, we choose these animals just to evaluate what's happened. We have some few animals that develop food addiction. So let's see what's happening in the brain of these animals. And in our early studies, we observed that in these animals that develop food addiction, there is an increase in the methylation of the Cb1 receptor in the prefrontal cortex. Remember that when we methylate a gene, we are going to inhibit the gene. So if we decrease methylation, we are going to produce the opposite, to increase. We are going to increase the expression of Cb1 receptor. And we are going to increase the amount of protein, or amount of Cb1 receptor in the prefrontal cortex. So those animals that develop food addiction has a higher concentration of Cb1 receptor in the prefrontal cortex. That was the result that I already presented last time. So now we have postulated the following hypothesis that we have tested. If we have more Cb1 receptor in the cortical areas, we postulate that the glutamatergic, the excitatory projection of the prefrontal cortex to the limbic system, they are going to be decreased. This activity is going to be decreased. So at the moment that you decrease this activity, we can postulate that we are going to have a failure in the inhibitory control of the cortex through the limbic system due to this overactivation of the endocannabinoid system. And if we lose this inhibitory control, of course, we are going to produce overeating despite the negative consequences, and we are going to produce this food addictive behavior. That was our hypothesis, but we must demonstrate this hypothesis. I mean, this is the result that I'm going to present now. So how we can demonstrate this hypothesis? Well, we must, the first thing that we did was to, in collaboration with Bea Luz in the University of Mainz, we obtained animals, conditional knockout animals that are deficient in Cb1 receptor only in these glutamatergic projections, only in the glutamatergic cells of the forebrain. So this animal has Cb1 receptor everywhere, like any normal animal, but lacks Cb1 receptor only in these glutamatergic projections of the forebrain. So in this animal, without Cb1 receptor here, we postulate that it doesn't have Cb1 receptor in these areas. We postulate that this animal will be resilient to develop food addiction because it doesn't have Cb1 receptor in this crucial area. And indeed, we did exactly the same protocol during 112 days in this case and evaluate the addictive behavior at the end of the training. In this conditional knockout animal that lack Cb1 receptor only in the glutamatergic projections. And what we observed, this animal just learn the behavior and press to obtain the chocolate pellet, but when we evaluate the addictive-like behavior at the end of the training, we observed that there was a very important decrease in all the criteria. There was a significant decrease in the persistent to response, in the motivation, and in the response after punishment, in the compulsivity. And look the addictive, the percentage of animals that become addicted. Wild-type animal, as in the previous experiment, 25%. The Cb1 knockout, the glue Cb1 knockout, only 6.9%. So there was a dramatic decrease in the development of food addiction in these animals. So why? Why there was this decrease? So if we postulate that there is a decrease in the activity in the cortical area, let's evaluate this activity. So we must evaluate you doing electrophysiological procedure. So with electrophysiology, we can evaluate the excitatory currents and the inhibitory currents. Those dependent on glutamate, those dependent on GABA. And of course, we evaluate the amplitude of the current and the frequency. And this is the results in the prefrontal cortex. So in the inhibitory activity, there was no modification at all. But look in the frequency of the excitatory activity. There was a decrease. So that means when you remove Cb1 receptor here, in the cortical area, you decrease the excitatory activity in the cortical areas. In the cortical areas, but exactly the same in the limbic areas, in the nucleus accumbens. In the nucleus accumbens, the same. You have a decrease, you have an increase in the excitatory activity in the nucleus accumbens, no modification in the inhibitory activity. So you modify, you enhance the excitatory currents as expected at the moment that you remove Cb1 receptor. You enhance excitatory currents, why? Because you increase glutamate release. This you can also demonstrate by electrophysiology. There is a pair pulse facilitation. That means you measure the capability to activate these glutamatergic neurons. And you have an enhancement of this capability. So that means that this animal that doesn't have Cb1 receptor in this area have an increase in the activity of the excitatory activity in this area and an increase in the release of glutamate in this area. And this is the mechanism that we postulate. We postulate as follows. We remove Cb1 receptor in these areas. What we do? We enhance glutamate in this projection from the cortex to the limbic system. This enhancement in glutamate, we activate GABAergic inhibition to the VTA, to the ventral tegmental area, which will decrease dopaminergic activity in the limbic system. So you have an enhancement in the activity in the cortical areas, a decreased dopamine activity in the limbic system. This is the perfect situation to control your behavior. The perfect situation to just make a rational control of the behavior. Decreasing cannabinoid activity in this cortico-limbic area. Well, if by decreasing Cb1 receptor you generate a resilient mice, we also postulate that we can do the opposite. If we just block the activity of this neuron, the glutamatergic activity in this neuron, we are going to produce a disinhibition. We are going to generate a mouse that is already added to food. At the moment that we just remove glutamatergic activity, we do the opposite. Before, we just increase glutamatergic activity by removing Cb1 receptor. Now, let's remove the glutamatergic activity. How we can remove the glutamatergic activity? We can remove the glutamatergic activity by using DREAD. DREAD is just an artificial receptor that we just put in the prefrontal cortex of our animal. This is an artificial receptor that we construct, and this receptor is just activated by an external compound. An external compound that is called CNO, the clozapine N oxide. And this external compound doesn't produce any effect, but just activate the receptor. And the receptor we can manipulate as we want. The receptor that we have incorporated here is an inhibitory receptor. So that means you just incorporate the receptor in the cell, and when you administer the compound, you are going to inhibit the cell. And the receptor, we incorporate it in these glutamatergic projections of the prelimbic system. So we administer the vector, we just express the receptor in all these areas, and let's see what's happening with the development of food addiction. So remember that the receptor is going to inhibit selectively this glutamatergic projection. And of course, in these animals, we have, the first is the verification that we have a decrease in the activity. Electrophysiologically, we can demonstrate this decrease in the activity. Look, the firing of the cortical neurons when we put, well, in baseline condition and after the administration of CNO, there is a decrease. But let's see what's happening with addiction. We expect to develop addiction. So let's make a shorter protocol. A protocol of only 4 weeks. 4 weeks is not enough to develop food addiction. Not enough, but look these animals. Look the wild-type animals. Animals without modification, you have 15%, few animals develop addiction. In animals that you generate this genetic manipulation, putting the inhibitory receptor in these cells, you have 42% of the animals that only after 4 weeks, you are able to develop food addiction. So we demonstrate that by inhibiting these particular pathways, we are able in a very short period, only 4 weeks, to develop food addiction. And just to finish, we have administered our vector in all the projection of the cortical areas. And remember, the cortical areas project to a lot of structures. We would like to dissect a little bit more which one of these areas was involved. So now the procedure is a little bit more complex, because from all these areas, you can imagine the one that was interesting for us was from the prelimbic to the nucleus accumbens. How we can manipulate selectively this area and only this area? By a double retro-DREAD approach. That is as follows. We administer our DREAD virus in the cortical areas. DREAD will be expressed only in those cells that contain a sequence that is called CRE, an artificial sequence. This artificial sequence we are going to administer in a retrovirus in the nucleus accumbens. So CRE will be transferred from the nucleus accumbens to the prelimbic area, to the cortex, just by this retro transport. So in this way, using this retrovirus, we are going to transmit CRE from the nucleus accumbens to the limbic system. We administer our drug in the prelimbic system, and it's going to be expressed only in those cells that contain CRE. Those cells that are sending the projection from the cortex to the nucleus accumbens. And in this way, we are going to have this receptor only in the projection from the prelimbic system to the nucleus accumbens. It's a complicated but very selective procedure to just investigate the activity of one particular pathway in the brain. So in this animal, at the moment that we give CNO, we are going to inhibit only this pathway, glutamatergic pathway from the prelimbic system to the nucleus accumbens. So we expect that this pathway is involved. And we did the same. We demonstrate by electrophysiological ways that this pathway was inhibited. But look what's happened when we develop food addiction. When we develop food addiction with this procedure, was very nice, because there was 50% of the animal that just in a period of 4 weeks developed food addiction. And for me, probably the most interesting is that we dissect which one of the behavior was mediated by this pathway. This pathway mediates mainly the compulsivity. This animal was unable to stop the behavior after the food shock. So it's a little bit complicated, but sophisticated way to dissect the role of a particular pathway, prelimbic nucleus accumbens, for one component of food addiction, that is compulsivity. So with this procedure, we demonstrate first that the Cb1 receptors are indeed involved in food addiction, that when we suppress Cb1 receptor from the glutamatergic medial prefrontal cortex area, we protect against the development of food addiction, and in the opposite way, we generate a mouse that is vulnerable to develop food addiction just by decreasing glutamatergic activity in the prefrontal cortex. And the particular pathway that is going from the prefrontal cortex to the nucleus accumbens seems crucial for food addiction, and mainly for the compulsive behavior related to food addiction. And just to thank all the, imagine that this is a collaborative study with the participation of several laboratories, Laura Domingo, Eric Saneabre, and Elena Martin in our lab, electrophysiological study by Mara Diersen, Eduardo Domínguez, and Mara Navarrete. Dread studies, we have the help of Raúl Landero, and Bea Luz and Íñigo Ruiz from the University of Mainz for the animals genetically modified. Thank you very much.
Le premier jour, Marc Auriacombe.
Well, Marc Auriacombe is going to give the most accurate potential definition of addiction. He said, well, it's the losing of control. And this definition you could demonstrate in this murine model by changing the glutaminergic pathway, and whether you activate or reduce that pathway, we do see that there's a losing of control. So you could demonstrate that direct connection. It's a very beautiful work that you presented right now. Do you have questions? At some point, you talked about ribonaban. It is true that we hoped at some point that we could reduce the general consumption of ribonaban. Did you find an interesting conclusion in your study about that? Because it's an antagonist of Cb1. Yes, indeed. In the first test that we conducted, I didn't show the slide, we developed a model where the mouse had an addiction to food, and then we administer ribonaban. And when we administer that drug, it completely stopped addiction. So there was a good correlation with the preliminary results, in which in human, we see that rimonabant does reduce very strongly the food intake. So that was very good for metabolic changes associated to obesity. Now, rimonabant was also tested in nicotine addiction, and we have a number of clinical studies where they demonstrate. There are 3 of them, one in Europe, one in the United States, and one worldwide. And in the United States, there was a significant decrease of addiction with rimonabant compared to placebo. So we do have models which demonstrate that in that type of addiction, and we could demonstrate that in an animal model for food addiction. Well, I'm a clinician. I'm very far away from research. So for you, this model that you just explained is very interesting. Do you think that, so for food, do you think what type of substance, what type of drug is it better mimicking? Because food initially is not a drug. It's a behavior that we see in our patient, which makes us think that it's a behavior based on addiction. So this model that you show, yeah, well, I agree with you. To begin with, food is not a drug. Now, what we see with that model, we're in the process of trying to identify the different mechanisms involved in a behavioral change. And we're really at the crossroads between food and drugs. So the behavior is changed. In the different tests that we made, we really focused on a specific compound, and we really focused on compulsivity, compulsion. So we are trying to identify a specific pathway in the prefrontal cortex, the accumbens nucleus, and we know it is involved in compulsion and addiction to food, and also addiction to drugs. So this is very similar in terms of mechanism. So I couldn't make a direct connection with one drug or the other. It's a notion that you will see all the drugs that have an impact on the prefrontal cortex. So that could be cocaine, that could be any type of drug. So it's just the same sort of mechanism that is at play.
Vous avez d'autres questions?
Are there other questions? If not, we are going to listen to the next and last presentation of this session dedicated to neurobiology, new therapeutical avenues. We would like to really thank Mr. Roque, Professor Roque, for being here. The moderator of the main presenter is Professor Bernard ROQUES. Professor Bernard ROQUES wrote a book on the dangers of drugs. It was published in 1998. He is a specialist in chemistry and neurobiologist, specialized in neuropeptides. It's an emeritus professor at the Paris Descartes University, and since November 1997, he's a member of the Academy of Sciences in Paris. He's going to discuss a new avenue which can help in reducing pain. So he's going to discuss new analgesic drugs by a complete inhibition of opioid enzymes.
Committee and scientific committee to invite me. It is always a pleasure to be in Biarritz for the THS. It's about the 10 times that I'll participate to this very interesting congress. And so I would like to present new data. And the title of my presentation is in French, but I give my talk in English and Franglish, probably. So this is the extension of the property, but you can see the... Okay, so may I have the next... Okay, so before discussing about the new results, I would like to present the strategy which has followed in my laboratory and in the startup that we have created with Marie-Claude Fournier-Zalewski, and the name of which is Pharmalids. So, you know, the strategy is a physiological strategy in which we have taken into account the presence of endorphin, different forms of peptide, which are released into the brain, but not only into the brain, but also in all parts of the body. As you can see, there is a... Can I use... You have 4 different peptides, beta-endorphin, which interact with the mu receptor. The most important for us is the enképhaline, the met and le enképhaline, which interact with both with mu and delta receptor, and dynorphin, which interact with the kappa receptor indicated. There is also 2 endomorphine, but they are not the subject of my talk. Okay, so j'appuie là-dessus, non? Non, mais qu'est-ce qu'il fout là, celle-là? It's only to show you that morphine is the most often used molecule able to relieve the pain sensation, and this is true for all the type of disease, of pain disease. But as you know, morphine has several side effects, in particular sedation, eventual constipation, and even a risk of addiction. And therefore, this is obviously morphine use is a pharmacological idea, whereas in our case, we have selected a physiological approach, which consists in potentiating the action of the internal substances, internal peptide, which was presented just before. This is only to show you, this is somatosensory, a schematic representation of the somatosensory system, in which we have the receptor which are in the different organs. For instance, oh, I'm sorry. So this is a schematic representation of the somatosensory system, in which the nociceptors are located in the different organs. For instance, in the muscle, you know. Ça va pas du tout. Okay. So in the muscle, in the different organs, and in which the stimulation of this nociceptor transmits the sensation to the spinal cord, and then the sensation is projected to the brain, to the different structure of the brain, and the brain examine and class the different stimulation as a sensitive stimulation or painful sensation. Okay, so our strategy was to use, just before, was to use the endogenous opioid system, and in this case, the 2-enképhaline, the meth and the le-enképhaline. These two peptides, neuropeptides, pentapeptides en fait, have been injected in the brain, directly in the brain, and have given a response analogous to that of morphine. But the time effect is very short because these two neuropeptides are inactivated by two enzymes, aminopeptidase N, which cleave between tyrosine and glycine, and neutral endopeptidase, which cleave between glycine and phenylalanine. And obviously given fragments which are inactive toward the mu and delta receptor. And therefore the idea is obviously to synthesize a compound able to protect from the degradation due to the action of the two enzymes. And this is the reason for which we use the name Denki's dual enképhalinase inhibitor. So on the next slide, you can see that we have developed two types of Denki's. And in this slide, we have the most efficient and the most powerful, which is a compound able to recognize the different parts of the two enzymes, the aminopeptidase. For the two compounds, because the active fragment is P254, able to interact with neutral endopeptidase and with the APN by different fragments of the molecule. And so, as you can see, the inhibitory potency is in the nanomolar range for the two enzymes, for NEP and for APN. So next slide. So this is also to show that there is a very close correlation, local correlation, between the two receptors, the mu and delta receptor, at the center of the slide, and on each part for neutral endopeptidase and APN. And this is clearly shown, this is clearly indicating that the system is present in different structures, both in the brain and in different organs, and for instance, in the bone, okay, and the spinal cord. So one of the most interesting control which has been done for the role, the physiological role of proenképhaline was the knockout of the preproenképhaline. In this case, the gene is enabled to form the final product, the final pentapeptide, meth and le-enképhaline. And we have shown in this case that you can, if you compare the results in the hot plate test, peut-être vous pouvez le montrer, vous. On appuie là. Là. You can see that in the hot plate test, you have a large increase in the response, indicating that the release enképhaline produce reduction of the pain. After the genetic ablation of the preproenképhaline, there is a strong reduction. You can see there. And now, if you use Denki's, in this case, this is RB101, at 2 milligrams per kilogram, In the control animal, you have a large increase in the jump latency, indicating an analgesic response, and this response decrease in the case of knockout animal, since you have not any peptide to be protected. Another control is the facility in producing by administration of denkies in the increase of enképhaline level in different structures of the brain. This is the case for EB101 in nucleus accumbens. You can see that after IP injection of 80 milligrams per kilogram of these denkies, there is a large increase in the concentration of enképhaline. And this is a metenképhaline. And in this case, this is another denkies, which also produce an increase in the periagudical gray matter. But the most interesting observation in this one, if you made this kind of manipulation with metenképhaline, which is given in a picogram per fraction of a perfused spinal cord by artificial CSF, you can see that without painful concentration, you have a control of about 4 picogram per fraction. And obviously, you have an increase by chelatorphan, which and another denkies, which protect the release enképhaline, metenképhaline. But you can see that the most interesting result is that when you have a muscle pinching is obviously very painful for the animal. In this case, you have an increase, obviously due to the painful control, and you can see that we have about twice increase in concentration of metenképhaline. And obviously, if you add chelatorphan, in both cases, you have about the same result, since you protect the enképhaline, which are released, in this case, by the painful stimulation. This slide shows a very important observation, which has been done by Alan North in United States. You can see that this is slices of locus ceruleus, and we perform control with metenképhaline here. And obviously, as expected, metenképhaline produces an hyperpolarization. And in presence of chelatorphan, which protect the level of metenképhaline produced, you have obviously, as expected, an increase in the response. But the most important result in this one, you can see that chelatorphan alone is unable to give hyperpolarization, and this obviously indicates that there is a very low basal release of enképhaline in this structure, and as locus ceruleus is involved in the control of respiration and cardiac rhythm, and at the source of physical symptoms of addiction, we have observed that denki have no detrimental effect on respiration and cardiac frequency, and this has been demonstrated by many people. So this is to show you that, as expected, for the similar affinity of the enkephaline and morphine, you have exactly the same response in such type of curve, which indicate that AD50 for morphine is 1.6 milligram per kilogram, and PL37 1.8 milligram per kilogram. This is only to show you that the expected strategy was confirmed by this type of experiment. Even more interesting is this one. This is a potentiation of the response of morphine, which is very low in this case. This is an analgesic response in the hot plate test. You can see that RB11 at increasing concentration give a response, which is in percentage of analgesia, around 40, whereas in this case, the milligram of kilogram by kilogram of morphine, 0.5, is unable to give a significant response, but the association at the different dose of morphine and RB11, the dual inhibitor RB11, you have an increase, a large increase in the response, and this is very important since we could hope to use this kind of experiment in different methods in palliative pain treatment, for instance. This is in due course actually at this time. So this is to present the results and to explain the result in neuropathic and inflammatory pain. In the case of neuropathic and inflammatory pain, there is a very large release of enkephalin at the level of the nociceptor, and this is due both to an attraction of lymphocyte to the site of the pain, and you can see there that the lymphocytes are recruited at the level of the injury, and obviously they interact with morphine receptor, with mu and delta receptor, and there is also mu and delta receptor expressed and transported at the level of the terminal coming from the ganglion, the spinal ganglion. So this is the result obtained in neuropathic pain, where PL37 was shown to reduce allodynia and thermal hyperalgesia. You can see that if you use the von Frey test, the filament, the different filaments of increasing dose, increasing force expressed in G, produce obviously pain. And there is a reduction of this pain sensation by PL37, PL37 at different doses. You can see that at low dose there is no real action, but at lower dose you have a reduction, which is shown by an increase in the G supported dose of von Frey test. And this is also shown in the plantar test, in which we have an increase, a decrease, I'm sorry, in the pore withdrawal in this model. And the effect of this PL37 is reversed by administration of naloxone methionine, which indicate that the action occurs at the periphery. So this is a series of slides which demonstrate the possibility to give not an additional but a potentiation of the effect of a combination of different substances. For instance, the denkies with THC. In this case, you can see the enormous protection of RB101 in presence of tetrahydrocannabinol, and you can see the enormous increase in the response percentage of analgesia. This is supported by the shock by experiment with shock and mice, and in this case, you can reduction in the RB101 and THC, which are associated to give a response which is antagonized by the Cb1 antagonist. So this is the plurality of pain relief by dual enképhalinase inhibitor. This is a summary. PL37 is active after oral administration of moderate inflammatory pain. For instance, due to the carrageenan administration, model of neuropathic pain, sciatic nerve ligation, c'est une collaboration with Rafael Maldonado. Streptozocin-induced model of diabetic neuropathic in collaboration with Alain Chalier in Clermont-Ferrand. And osteosarcome-induced thermal hyperalgesia in collaboration with another Spain friend, Anna Bahamonde. So the synergetic effect controlled by isobolographic analysis for all the models with different molecules acting on different opioid receptors are shown on this. So we observe a synergetic effect involving interaction between cannabinoid and opioid receptor system. This is true for the Cb1 receptor, for the Cb2, but also for the, this is for the Cb1. You can see the synergetic effect. For the Cb2, this is exactly the same type of result. Okay, and also for the inhibitor FAH, URB937, in which you have a very large synergetic effect. You can see the addition of the two substances without synergetic effect and the result after synergetic potentiation. This is only a table in which you have the experimental concentration ED50 expressed in milligram per kilogram, and you can compare the theoretical ED50, which is shown here, and the real experimental values, which show clearly a very high increase in the data. So this is proof of concept in phase 2 studies in patients in which they are in which they have pregabalin treatment, and this is pregabalin placebo and pregabalin plus PL37. You can see a large decrease in the evaluation of the pain score for the patient treated by pregabalin and pregabalin and PL37. All results are presented in different congresses, and especially in the YASP Congress. So this multi-target-based anti-nocivity strategy is in combination with UNM regarding enzyme of various compounds with direct and indirect mechanism of action with opioid system provide therapeutic advantage in terms of efficacy and safety by allowing the reduction of dose of one of the compound or of both, which is of the utmost interest in the chronic treatment of cancer pain, as discussed for the morphine in palliative pain, for instance. So this is the result obtained in clinical phase 1 security pharmacology, in which 137 subjects, comment ça se dit déjà, c'est des volontaires, have received PL37 by oral dose by increasing dose between 6.25 milligram until 100,000 milligram, and multiple dose between 4 times per day during 5 days. And you can observe no depressive respiratory effect. Sorry. Oh, merde. Je reviens en arrière, le nom de Dieu. Merci. No respiratory depression, no cardiovascular effect, no constipation even after administration chronic, no tolerance after repeated administration, no nauseous in the people and in the animal model, and okay. So this is the second part of my presentation. The opioid receptor delta and their ligands are present in high quantity in the epithelial cells and are implicated in their protection. And these studies have been done in collaboration with Institut de la Vision, l'hôpital Saint-Antoine, directed by, je ne me souviens plus de son nom d'un seul coup. C'est pas grave. So, it has been shown that another dengue, PL265, is able to diminish the pain in various lesions of the cornea, of the corneus lesion, and the local administration of this dengue significantly reduce the inflammation of the corneus. And all these effects are obtained by local way, and they don't use the central nervous system. So the last part is the effect of this dual inhibitor on the migraine crisis. So the migraine crisis is probably due this is the last hypothesis, to a large electrical storm coming from the hypothalamus. And this produces a large increase in NO and in CGRP, calcitonin gene-related peptide, which traffic from the C fiber to the Al-delta fiber, and this potentiates the response. And on the other one, there is NO, which activates the main ingenial artery, and this obviously produces direct vasodilatation and activate signaling cascade, resulting in activation of NO production. And NO is able to cause vasodilatation and diffuse to the trigeminal nerve terminal, where it enhances the further relief of CGRP. So this is an increase in the potentiation of the migraine crisis. So different experiments have been performed by, in close collaboration with Radwan Dalel, a very good specialist of the question in Clermont-Ferrand. And you can see that in this slide, we have an absence of reduction in the effect of PL37 after repeated oral administration of PL37, which show that this is saline, which obviously produce an increase in the filament, von Frey filament pain. And you can see that PL37 reduces completely this sensitivity to the von Frey filament. So this is a comparison with sumatriptan, which is the usual treatment of migraine, with PL37 and vehicle. And you can see that in the case of vehicle, you have a large sensitivity to von Frey filament with a strong reduction of the supported dose expressed in G. You can see the result here. And you can see that PL37 produces a large difference, and the so this slide suggests a preventive action of the NENKI, of the induced migraine crisis, since this appears after a large time in minutes. So all these results are summarized in Nature Research Drug Discovery, and if you are interested by all these results, you can look at this paper, which has been published in 2012, and even for the last result. So thank you for your attention.
Y a-t-il des questions dans la salle?
Are there any questions? I would like to ask you, when will these molecules be available to be used for regular use?
The clinical specialist, and for the neuropathic pain, we have a second series of experiments with the last d'Enkees, 265 là. Et pour le premier, on pense qu'on devrait avoir une phase 3 dès qu'on va toucher.
We think we should have a phase 3 because to have a phase 3, we need I don't know how many $1 million. And it's more complicated, but I think that we can reasonably hope that this strategy will be productive, will be profitable in 3 or 4 years, which will be a victory. Rafael does know it. And what was the pregabalin dose used in the previous phase? There was pregabalin in association. There was a dose of pregabalin used. $40 milligrams. If there are no further questions, we're going to stop for now. We thank all the 3 speakers. And we're going to thank you for your presence all the way to the end.